Hello, everyone, and welcome to today's live broadcast, Deciphering the Complexities of Neurodegeneration and Neuroinflammation with Gene Expression Profiling, presented by Kelly Miller, Application Scientist, NanoString Technologies, Incorporated. I'm Alexis Krause of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by NanoString Technologies. For more information about our sponsor, please visit www.nanostring.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Dr. Miller will be responding to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Today's webcast will be available for on-demand viewing through June of 2018. Please join me in welcoming Dr. Kelly Miller. I will now turn the presentation over to her.
Good morning, everyone, and thank you for joining our webinar today. Really excited to go over some of the content here. Before I get started, I would like to remind you that this is an interactive webinar, so there's a Q&A box on the right-hand side. Definitely feel free to ask questions throughout the presentation, and we'll try to answer them as quickly as we can, and we can also answer at the end and get some feedback as we go along. So this webinar is on deciphering the complexities of neurodegeneration and neuroinflammation with gene expression profiling. And I'm Kyle Downey. I'm a product manager at NanoString, and I'd like to introduce you to Kelly Miller, who's going to give the talk today. She has a Ph.D. in Biomedical Science from the University of Florida. After that, she did her postdoc at Charité Medical University in Germany. She was a research scientist at UPenn Center for Neurodegenerative Disease Research, and most recently, she joined NanoString as a field applications scientist. So Kelly, take it away.
Thanks so much for the introduction, Kyle. As mentioned, prior to joining NanoString, I was an academic neuroscience researcher myself, so I'm really very excited to be introducing NanoString's first neuroscience-specific products in this webinar, our neuropathology and neuroinflammation gene expression panels. So I'm going to spend the next 40 minutes or so giving an overview of how NanoString technology works, demonstrating performance of the platform on relevant sample types, describing our new panel content from the neuroscience field, and sharing some really interesting data generated using that neuropathology panel. So I hope that by the end of the presentation, you have a solid understanding of how the technology works and is suited to your particular samples, and therefore how you can use these tools that I'll be presenting today to move your own studies forward. The basis of NanoString technology lies in the nCounter instruments combined with a unique barcoding chemistry that together allow for the simultaneous digital quantification of up to 800 targets in a given sample. And NanoString started as an RNA company providing solutions for gene expression profiling, but the technology has been adapted to allow for expanded applications that include not only mRNA profiling, but also microRNA, fusion gene detection, DNA profiling with applications like copy number variation and single nucleotide variant detection, and even protein analysis. And this happens all on the same instrument with the same readout using the same chemistry. We offer hardware solutions for both investigational and clinical use with 3 different instrument options, and also assays suited for any type of project, whether you aim for targeted discovery using one of our pre-curated gene expression panels, multi-analyte analysis with our 3D biology assays, which allow you to simultaneously profile any combination of RNA, DNA, and protein in a given sample, or if you want to perform sample multiplexing with our PlexSet assays. No matter what assays you're using, we utilize one core chemistry, and the basis for these applications lies in simple base pairing. The platform is measuring individual nucleic acid molecules, hybridized to fluorescent barcodes, allowing you to perform single or multi-analyte analysis or even morphology-driven profiling with our new digital spatial profiling application, which is our latest initiative to utilize the nCounter chemistry to extend our profiling capabilities to fixed tissues in a spatially resolved manner. And this is technology that's sure to prove exceptionally useful in the field of neuroscience in particular. So I mentioned our pre-curated gene expression panels. I'd like to go ahead and introduce both our neuroinflammation and pathology panels so you can begin to understand what types of questions you can address in your samples using these tools. So I'll go through and review the content and the important features of both panels. Start the neuropathology panels. Here we've designed a panel for human and one for mouse. Each contains 770 target genes, including 10 reference genes that were carefully chosen by taking into account the stability of expression across a wide range of neuropathological conditions. We also build in a series of positive and negative controls into every assay on top of the 770 mentioned genes. And for analysis purposes, we provide functionally annotated gene lists for all of the content, which is broken down into 6 fundamental themes intended to really broadly address key topics in neurodegenerative disease research, including neurotransmission, neuron-glia interactions, neuroplasticity, structural integrity, neuroinflammation, and metabolism. So in total, this content covers 23 different pathways and processes, and it was selected by reviewing current literature in the field and also consulting with researchers at a variety of academic institutions, biotechs, and pharmaceutical companies. And finally, amidst the gene content, there are also cell type specific gene signatures built in to allow for cell profiling during the data analysis. You can see the 6 themes that I just mentioned broken down further. And if you look at the annotation column over towards the right, you'll see a more detailed description of categorical content, including the number of genes representing each particular pathway or function. One category I want to point out is neuroinflammation, which is a process that's highly relevant for neurodegenerative diseases like Alzheimer's and Parkinson's, but is also relevant for nearly all neuropathological conditions. So you can see on the right-hand side that there are nearly 100 genes devoted just to monitoring the functional state of microglia, as well as another approximately 50 cytokines that are all profiled on this panel. Also, in terms of neuroinflammation, what's not listed there are a number of astrocytic genes that have been included, which allow for the monitoring of astrocytic responses as well. And here you can see a table that I took from one of Michael Safraniev's reviews describing different functional categories of genes modulated by reactive astrocytes. And I've highlighted those genes that are covered by this panel in bold. So overall, you can see that there is very good coverage of mechanisms underlying neuroinflammatory processes, even though this panel more broadly covers neurodegenerative disease pathways, much more so than our neuroinflammation panel. Another category I want to highlight just below neuroinflammation is metabolism, which includes nearly 50 genes devoted to monitoring the unfolded protein response, almost 100 for oxidative stress, and more than 30 for autophagy, which could prove really valuable for, say, Parkinson's research, among others. The last feature of the panel that I want to point out is the cell type profiling feature. Here you can see a few key neural cell types, and to the right you see genes whose expression has been empirically determined to be largely specific to those certain neural cell populations. So our nSolver Advanced Analysis software module will use these genes to measure the abundance of these cell types or their reactivity in the case of, say, activated astrocytes, for example, thus allowing you to assess cell-specific enrichment in your heterogeneous samples. Moving on to the neuroinflammation panel, you can see that this panel is also designed for both human and mouse and also targets 770 genes, which includes 13 reference genes. And again, this panel comes with a fully annotated gene list, and the content is broken down, in this case, into 3 core themes that were selected to allow for analysis of key aspects of neuroimmune interactions and also to comprehensively evaluate pathways and processes that are associated with neuroinflammation. So in total, the content covers 23 different pathways and includes the cell type-specific gene signatures for 19 different cell types. The content here is broken down to cover immunity and inflammation, including both adaptive and innate immune responses, with a total of nearly 275 genes devoted to those processes, more than 100 genes devoted to cytokine signaling, 185 to microglial activation, and more than 100 to inflammatory signaling. In the neurobiology and neuropathology section, you can see the panel covers neuron, oligodendrocyte, and astrocyte function as well, with 81 genes devoted to neurons, 27 for oligos, and 55 specific for astrocytes, among, of course, the other categories listed here. And finally, you can see we have genes covering metabolism and cell stress, which include more than 200 targets. As with the neuropathology panel, the cell profiling feature is also included in this panel, with signatures built in for the 5 neural cell types and 14 peripheral immune cell types. And if you're interested in learning more about how these signatures are developed and how they work, you can check out this publication from our team. Now that we've reviewed the panel content and benefits, I want to share some performance data that I think illustrates the point I made in the beginning, that this platform is especially well suited to handle neuroscience samples, which often consists of small samples either from sorted cell populations or iPSC-derived neurons, laser capture samples, or microdissected mouse brain regions, for example, and also from post-mortem human brain tissue that's not only fixed, but often paraffin embedded and therefore highly degraded. So since there aren't really a lot of robust solutions for some of these samples, NanoString offers the possibility to gain a lot of otherwise potentially unattainable information. The first example I have here is from a 2012 Nature Medicine paper, and here they're demonstrating the reproducibility obtained from very poor quality FFPE samples. If you're familiar with bioanalyzer traces, you can see that the quality of these samples is really, really bad. And they used as low as 25 nanogram input. And yet, you can see in the graph on the left, intercorrelate replicates all with R-square values above 0.99. Specifically, each point on the graph represents the correlation from an independent pair of samples, showing that the system is highly reproducible even when working with these degraded samples. The next example also focuses on fixed samples, but these are formalin-fixed cells that were processed for FACS. So on the top in the bioanalyzer traces, you can see the effects of fixation with unfixed cells on the left and the fixed cells on the right. And in this study, the authors compared qPCR and NanoString. The data on this slide are the qPCR data, and what we're looking at are the delta CT between fixed and unfixed cells for 2 different genes, 18S and GAPDH, among 3 different cell types. So obviously, we'd all like to see a delta CT of 0 to be sure that our technical procedures weren't impacting our biological readout. But in this case, you can see that in 5 of 6 instances, fixation led to a significant difference in gene expression as measured by the qPCR. Now, these figures, they show the NanoString results, and you can see firstly that they've multiplexed several genes. And on the left, you see the actual data counts for each gene compared between the fixed and unfixed cells with minimal differences. And on the right, you can see that there's a correspondingly high correlation between counts. And the authors concluded in this study that NanoString, but not qPCR, proved equally efficient in profiling those fixed and unfixed samples. Similarly, this data was generated using our neuropathology gene expression panel, the human panel, and it shows similar reproducibility between fresh and FFPE tissues. So in these scatter plots, you're seeing the correlation between fresh frozen and FFPE brain tissue taken at autopsy from 2 different patients. And you'll see that there's an excellent correlation between the fresh and FFPE samples. And what's even more impressive about those R-square values is that the fresh and FFPE material were taken from opposite brain hemispheres, which certainly introduces variability in gene expression already. So these values are really quite good. And below this, you can see the counts per gene from a randomly selected subset of targets on the panel, demonstrating what we saw in that previous study, which is that counts are consistent between samples or preservation methods. The last set of performance data I want to show you speaks to sample flexibility. Because NanoString doesn't require amplification or enzymes, you can work with whole RNA or directly with cell lysates. And this slide shows how the data compare between 40 nanogram of total RNA and 4,000 lyse cells. And you can see that the lysates performed equally well. And here's evidence from the literature where they make a similar comparison using either 100 nanogram total RNA or 10,000 lyse cells. And here they compare data from multiple donors at different time points, and in all cases, there was a very high correlation between the results obtained. Really, again, just demonstrating that this technology presents a viable option for samples that consist of precious, low-yield cells in which maybe you don't want to or can't extract RNA, or projects in which the sample number may make RNA isolation impractical. Here's a similar comparison showing here, in this case, FFPE lysate performance for brain samples. So this includes both purified RNA from FFPE in green and crude FFPE lysate in orange, both compared then to purified RNA from fresh frozen material. And this really shows, again, this equivalent performance of the FFPE RNA and FFPE lysate. I'll just share a couple more pieces of performance data that were generated using our neuropathology panel. So firstly, in considering common sample types, because this platform is so amenable to FFPE, we expect a lot of translational studies that could serve to connect the data obtained in the past from studies in animal models on over to the human setting. And for this purpose, post-mortem FFPE tissue is certainly the most readily available human brain material. So I already showed you earlier the excellent performance of FFPE material compared to fresh frozen using the NeuroPath panel. And here we wanted to test the performance of these samples in terms of post-mortem interval. So in this heat map of raw data, which is shown as data counts, you can see that an unbiased clustering approach demonstrates that all samples performed well and equally independent of the post-mortem interval. And that's really most evident when you look at the grouping at the top, and you see that these samples aren't clustering based on the PMI. So samples with a PMI greater than 35 hours can be found right next to samples that have a PMI of 6 to 10 hours, for example. Another important sample quality issue that we wanted to address is the performance of blood samples on the neuropathology panel. So unlike the neuroinflammation panel, which has gene content for peripheral immune cells, the neuropathology panel is, of course, more strictly CNS focused, and therefore we wanted to assess whether a substantial proportion of genes on the panel are detectable in blood. And as you can see in the heat map, again, showing raw data in which we profiled blood from controls and from patients with frontotemporal dementia, most targets were detected in a robust range, which you can see shown in orange. And overall, about 63% of all targets were detectable, confirming that this panel is reasonable for studies in which one might want to assess blood-brain correlates, for example. And now, just before I move on to share some of the biology uncovered with this panel, I want to take a minute to acknowledge our collaborators who worked with us experiments that I'll be mentioning and who provided the samples and are continuing to pursue these studies. All data you'll see today were generated using samples obtained from the University of Pennsylvania Center for Neurodegenerative Disease Research, which is led by Virginia Lee and John Trujanowski. And our partners in these studies include Stefan Prokop, Viviana van Dierlen, Hao He, and Unran Su. So obviously we're very grateful for the chance to work with all of them. The samples that we use to generate the data that you'll see today include both mouse and human material. For the mouse studies, we worked with tissue from 5X-FAD mice, while from the human studies, we profiled samples that came from Alzheimer's, Parkinson's, and FTD patients. And in all cases, we used purified RNA, and while the mouse tissue was fresh frozen, all of the human data I'll share were generated using FFPE tissue. Also, all of the data you'll see here were generated using our nSolver advanced analysis software, and again, all samples were run on either the mouse or the human neuropathology panel. So first I want to share some findings uncovered when we analyzed whole tissue samples from the 5X-FAD mice, which are a common AD mouse model. Again, these figures were generated using our nSolver advanced analysis module, and here you can see a couple examples of gene set-specific heat maps generated with unbiased clustering that show a clear segregation of expression patterns in the 5X-FAD mice compared to controls. So on the left, you can see that genes associated with autophagy are elevated in the disease setting. And on the right, you can appreciate increased expression of genes associated with microglial activation, which all make a lot of sense given what we know in the field about this model. And if you look at the cell profiling feature and focus on the microglial score in particular, you can see that this analysis demonstrates an enrichment in microglia that's right in line with what we see in that heat map. Looking at other cell profiling scores, here you can see a lower neuron score in diseased mice, which corresponds really nicely to the no neuronal loss that occurs in this mouse line. We compared pathology-related genetic changes in the mouse model to the human disease. We found similar pathways affected, such as vesicular trafficking, which you can see on the right is reduced in AD mice compared to controls, and also in hippocampal specimens from high AD patients, which you see on the far left-hand side. Looking more closely at data generated from the human samples, here you can see a heat map of the entire 770 gene set on the left and a blow-up of the outlined area to the right. And if you look at the order of the samples and the corresponding gene expression, you can see that this particular subset of genes undergoes a progressive decrease in expression that corresponds to increased disease severity, with controls at the top working down through low and intermediate AD cases to high AD cases. And these were classified using the ABC criteria. And I think that this data really nicely demonstrates that the content on this panel is relevant for profiling neurodegeneration, and it's really targeting pathways and processes that are changing in disease. An illustration of that point is seen here. So here we're looking at the pathway scores produced in NSolver, and we found a conserved disturbance in neurotransmission across neurotransmitter response, synthesis, storage and release in high AD cases, again, with progressive changes evident in accordance with disease severity. So this was really very interesting and also reassuring in terms of detecting changes that really make sense and to which we can assign some biological meaning. Here you can see two plots from mouse models in which the animal model is mimicking the changes we see in the human samples. In all of these graphs, we're looking at transmitter response and reuptake. And in the center, you can see reduced scores for 5XFAD mice that were injected with human tau and therefore have more robust pathology than uninjected 5XFAD mice. And on the right, we again see that in the disease setting compared to control, genes associated with neurotransmission are again reduced. So overall, this data shows us that these changes we're observing are progressive, and most importantly, that some of these very important changes that we see happening in the human brain are also occurring in our animal models, which is really critical validation of these model systems. So here, looking at cell profiling scores in human AD, we can see something similar to what we saw in the 5XFAD mice, which is that microglia and astrocytes are enriched in association with high AD, and that neuron loss is occurring in the most severe cases. And here you can see that this microglial activation that we've detected genetically corresponds nicely to what was found using immunohistochemistry. Now look across diseases, and here we're comparing human AD hippocampus to hippocampus from Parkinson's patients at a late stage of disease. We can see some alterations that are conserved across different diseases as compared to the control patient. So in this case, you can see on the left that the unfolded protein response, which is obviously important for maintaining proteostasis, is reduced in both AD and PD, while on the right, you can see that chromatin modification seems to be elevated in both conditions, highlighting some shared mechanisms that may be important for pathogenesis and treatment across multiple neurodegenerative conditions. Here, as I also showed you in the context of Alzheimer's, we profiled midbrain specimens from Parkinson's patients and controls, and you see in these heat maps that specific subsets of genes are clearly differentially expressed in PD patients versus controls. So on the left, you can see a set of genes upregulated in PD cases, while on the right, you see targets that are reduced in the PD setting. So again, this highlights that the curated content on the panel is capturing disease-related changes and can really provide insights into disease mechanisms. And finally, here you can see with an unbiased clustering approach in the PD midbrain, there are clear differences in the expression of genes associated with key pathways of transmitter release and vesicle trafficking, suggesting deficits in neurotransmission, which is well in line with what we know about the pathogenesis and progression of PD. So this is the last of the data that I'm going to share. I just wanted to give a glimpse of what we've explored in this rich sample pool of AD, PD, and mouse samples that we obtained from our collaborators at Penn. Obviously, there are much more in-depth analyses yet to be performed, and that's what the researchers at Penn will continue working on. So we're certainly very excited to see the final conclusions of their studies. Throughout this presentation, you've seen that NanoString shows robust performance on a variety of sample types, including post-mortem human tissue, and that using our pre-curated gene content, you can rapidly identify biology that's differentiating normal samples, whether it's patient or mouse samples, from those of diseased patients or mice using our advanced analysis software. Moreover, we discovered conserved changes in human and mouse samples, demonstrating that these panels really enable a seamless translational research with reproducible results. And another great feature is the integrated cell signature content and analysis capabilities that enable that cell type profiling. And lastly, this panel content is designed to cover neurodegenerative diseases broadly, which really enables discovery of targetable biology that's consistent and that's relevant across multiple disease types. So I hope that this data and information that I've shared has helped you to envision how you can best address your research questions and also get the most information from your samples. And so we'd love to see what you all can discover using these panels. So we're offering a grant contest that will award 5 winners with 24 reactions of either the neuropathology gene expression panel or the neuroinflammation panel. The deadline for submission is March 16th, so I encourage you to go to our website and submit your application. Having demonstrated the type of interesting biological insight that can be gained using these panels and our platform, I just want to return again to highlight the simplicity and ease by which one can acquire this data. No matter what assays you're using, we're relying on simple base pairing for single or multi-analyte analysis or morphology-driven profiling. And it's the simplicity underlying these applications that lends itself so well to translational research. Because it's simply hybridization-based, gene expression analysis is performed without any amplification, cDNA synthesis, or use of enzymes, and therefore the system is really very tolerant of poor quality material or typically hard to profile material like FFPE, for example, making it really feasible to translate your data from, say, animal models to post-mortem human tissue, as you just saw. The technology is also highly reproducible, which again is tied to the fact that there's no amplification bias or reliance upon enzymes. And also due to the multiplexing capability, NanoString is ideal for pathway analysis, such as you saw in that data today, which really facilitates a more holistic approach to analysis. And finally, the workflow is really simple. So whether you're analyzing a few or many hundreds of targets, due to the fact that there's minimal hands-on manipulation, which certainly also contributes to the precision and reliability. And now I'll close by announcing an exciting program that will be available soon, which is our Digital Spatial Profiling Technology Access Program, which will bring the advancements of DSP to your project with the possibility of running samples in our labs in Seattle before the instrument is available on the market. This technology allows you to quantify multiple protein targets simultaneously on FFPE tissue sections on a slide in a completely non-destructive manner. So it provides you with quantitative digital spatial information. And we'll be launching our neuroprotein panel in April, and we'll start accepting projects at that time. And for more information about that program, you can email tap@nanostring.com. The address is shown here on the screen. And of course, please don't forget to go to our website and submit your neuroscience grant applications. So with that, I'll close and thank you all for your attention.
Great, thank you, Kelly. That was really good information, and there's a lot of content to unpack, so really appreciate you taking the time to go through all of that. I'd like to remind everyone in the audience that, again, this is an interactive session, so would really like to see if you have any questions that you'd like to ask Kelly, or if anything that has come up that you'd like a little bit more information about. So go ahead and send any of those into the Q&A box, and I'll go ahead and review those. It looks like there's a couple of good ones that have come in so far. So Kelly, first one that I'll ask for you. This is a good question. So how much RNA is required for the NanoString assay?
So the RNA input really depends on your sample type and what genes you're looking at or which panel you're working with. But in general, the range is anywhere from 25 nanogram to 300 nanogram. I would say our standard input for these neuro panels is 50 to 100. And I can say already that I've seen really robust counts coming off of these sample types on these panels. So, you know, for very, in a very general sense, I would say 50 nanogram is a great starting point.
Okay. And another question, kind of related, how does the NanoString assay perform on CSF as a sample type?
So CSF is definitely works on this platform. We have a publication on our website profiling microRNA and CSF. It worked great. We haven't run it specifically on the neuro panel, but again, in terms of input for CSF, that's something that you would want to optimize on a project by project basis.
Okay, great. All right, another one here. Can you describe in more detail how the cell profiling feature works with this panel?
Yeah, sure. So this cell profiling feature, and if you're interested in this, I would again encourage you to check out that publication by our team. The first author is Patrick Danaher, and there he describes the method that's used to measure the abundance of these cell populations. So basically it quantifies cell populations using marker genes that are expressed stably and specifically in given cell types. So these marker genes will act as reference genes specific to those individual cell types, and they should only be expressed in that particular cell type at the same level in each cell. So our advanced analysis software will perform all of that scoring for you, and of course it's taking into consideration those things that I just mentioned.
Perfect, thank you. And definitely want to remind everyone again, you can always add your questions to the queue. Okay, another question that came in, how were the genes in this panel selected?
Yeah, so for both of these panels, the genes were selected after, a really careful review of the literature, what's going on in the field currently, and then really going out and talking to researchers. As I said, we reached out to people not only in academia, but also in biotech and pharma companies, researched relevant databases, and known data that's online to really pull out the most relevant information for, in the case of the neuropathology panel, for all of the common neurodegenerative diseases, and really try and find the content that is specifically relevant and well suited to what seems to be going on in these diseases. And I think that was really the message that comes across in this data as we start to look at it. It was true for the mouse samples, for the AD, for the PD samples, that we can really segregate these samples, the disease cases from the controls, and we can really pull out pathways and processes that are being affected in disease. So I think in that regard, we really accomplished that goal.
Perfect. And here's a good question. So, can the gene content be customized? If someone has a favorite gene, can they add it in?
Yeah, absolutely. Both of these panels, you can add up to 30 genes to both of these panels. You can customize in that regard. Obviously, you can build completely custom panels at NanoString, but if you want to work with the content on these panels and just need to add in a few of your favorites or targets that may not be there that are particularly important for your project, then you can just add those on and you can profile a total of up to 800 genes.
Perfect. And another question on the cell profiling. Does the neuroinflammation panel have the same cell profiling feature? Is it any different?
Yeah, it absolutely has the same features, but in addition to the 5 CNS-specific cell types that are profiled on the neuropathology panel, it also contains 14 signatures for peripheral immune cell types. So it's working in the same way, but there's a lot more signatures built in.